Sorry. Today, I'd like to introduce Stripenn that detects architecture stripe from the chromatin conformation data using computer vision. Here is the QR code for Stripenn bioRxiv paper, so you can easily refer to the Stripenn paper using this QR code. First, I'd like to introduce architecture stripes. This is an example high-C heat map, and here, this linear structure is architecture stripe. This is a pattern where single locus form frequent contact with contiguous genomic regions. So I prepared a simple video for stripe formation. Here, this red spot is stripe anchor, and this stripe anchor is keep interacting with other stripe domain to form a stripes. So this structure was first predicted by Gutenberg in 2016 using polymer simulation. And it was found in activated V cell and named as architecture stripes by Bienetto in 2018. And these stripes are enriched in active spur enhancers and affects the transcription, and stripe anchors, this starting point of the stripes, are hotspot for DNA damage. Besides that, there are some interesting features of the stripe. First, stripe is formed in tissue-specific manner. For example, this stripe is formed by inverting the EPH84 domain in the mouse limbus. However, this stripe is not observed in this mouse embryonic stem cell. So we can say stripe is formed in the tissue-specific manner. And also, stripe formation can enhance the transcription. For example, in the mouse B cell cancer tumor, this stripe is formed, and there is a MYC and PBT located here. And when this super anchor of this stripe is deleted by deleting the CTCF here, these stripe domains are disappear, and the transcription level of the MYC and PBT is greatly reduced. And also, there are so many questions related to stripes, from the formation of stripes to the shape of stripes and the biological function of the stripes. So to address these questions, basically we need an accurate stripe colors. There are existing stripe colors. First one is Zebra, which was developed by Bienetto in 2018. It uses Poisson statistic-based method to detect the architecture stripes. It is very sensitive, so it detects a lot of candidate stripes. However, sometimes it detects this loop structure as a stripe, which I say false positive. And there is no scoring system for stripes in Zebra, and this Zebra original program is not publicly available. So from Feng US group, stripe color was developed using the Zebra algorithm. However, when I run this program, I found that there are many false positive codes, like as shown in this figure. And also there are some other stripe colors called domain class by R and chromocytes. Domain class by R detects loop and stripes at the TAD boundary. So the stripes within the TAD is not detected by the domain class by R. And chromocytes detects loop, stripes, and other features in high-C data using computer vision. However, when it reports the location of stripes, it gives only 1 point, like this, like shown in this figure. So it is hard to know the exact coordinate of the stripes. So as seen in the previous slide, there are technical gap for the stripe color. We need more accurate stripe color. So that's why I developed this Stripenn. Stripenn is an image processing based method implemented in Python. It provides stripe coordinates, and it has two scoring systems for stripes. And it is publicly available in GitHub and PyPy. Using any types of chromatin conformation data like high C, high chip like Plexic, and micro C, you can detect stripes. And this is the workflow of the Stripenn. The Stripenn workflow is divided into two parts. The first part is a candidate stripe search with image processing technique. And second part is the scoring stripe part using p-value and stripiness. In the candidate stripe search part, the Canny edge detection method is the key step. Canny edge detection is a kind of edge detection method that was developed by John Canny in 1986. So still it is very popularly used because of its low error rate and optimal localization and only one strong signal detection. So I applied this Canny edge detection to the high C data to detect this long and thin structure, the stripe. After we find candidate stripes using the imaging processing technique, we filter the candidate stripes using the p-value and then sort them based on the stripiness, which is a quantitative measure of the stripes. This is the stripiness measure, and it considers the contrast between stripes and their neighbors and the pixel continuity along the stripe domain and the median pixel value within the stripes. And we found that this Stripenn is outperform other existing stripe colors as shown in this pilot plot analysis. And we also found that the stripiness and average intensity of the Stripenn specific stripes are significantly higher than that of zebra-specific stripes. Here, I didn't include the chromosite and domain classifier specific stripes because they didn't give the exact stripe coordinates. Using this stripe, now we can do systematically analyze the stripes. So from now on, I will show you some data about these 5 questions regarding the stripes. The first one is, how is the distribution of the structural protein on the stripes? The second question is that, where are the stripes located? And third, what are the differences of the Stripy compared to the non-Stripy? And fourth is, are all the genes on the stripe highly expressed? And the last question is, are the genes on the stripe related to the cell identity determination? So for the first questions, using high C of the activated B cell in mice, we found that CTCF and cohesion subunit like SMC3 and RA21 were highly initiated at the anchor of the stripe. Here, the anchor of the stripe means that the starting point of the stripes. This data is consistent with the previous finding from Bianetto, and we also found that the recruitment of NIPBL is somewhat showing somewhat different pattern with others. The recruitment of NIPBL is distributed all over the stripe domain, but is still skewed to the anchor sites. And for the second question, we found that most stripes, more than 80% of the stripes, were existing in only a compartment, which is transcriptionally active and located interior of the nucleus. Considering that only 44% of the tags were existing only A compartments, we can say it is a unique feature of the stripe. Next, we compare the stripy tag versus non-stripy tag in A compartment. Here, stripy tag means the tag that includes the stripe, like this, and non-striped tag does not include any possible stripes. By comparing the stripy tag, which is blue, and non-striped tag, which is black, we found that cohesin subunit, the SMC3 and Ra21 are more actively loaded on the stripy tag compared to the non-striped tag. And also, we could see very striking difference in NPL. So this data suggests that NPL has very important role in the stripe formation. This data also suggests that cohesin is required to form a stripes. This data is HCT116 cell line. Here, when Ra21, which is cohesin subunit, is depleted, all the clear stripes are disappear. And this data is also consistent with previous finding that cohesin and its ATPase activity is required to form a loop extrusion. Next, I focused on the gene expression in stripy and non-stripy tags. When I compare the gene expression in non-stripy tag and stripy tags, I found there is no significant difference between them. Most genes within stripy tags and stripy and non-stripy tags were lowly expressed. However, when I compared the most highly expressed genes within the stripy tag and non-stripy tag, I found that stripy tag genes were significantly more highly expressed than non-stripy tag genes. Then why such behavior is observed? I found the answer from the active enhancer distribution, which is demarcated by H3K27 acetylation. So this data shows that the active enhancer is biased to the most highly expressed genes than other genes, and especially in stripy tag genes. This is one example stripy tag, including this stripe. This tag includes so many genes here, but when we see this RNA-seq track, we can find that only this CD247 gene is highly expressed along this stripy lesion. And we can also see that K27 acetyl, which is active enhancer marks, is biased to this gene body. So it shows that the active enhancer is biased to only one or a few genes within the stripy tag, and that affects the transcription. And since the CD247 is a surface protein of the T cell, our next question was whether this most highly expressed gene within the stripy domain are related to cell identity determination. So we performed the gene set enrichment analysis for non-stripy tag and stripy tag tags most highly expressed gene and found that these stripy tag genes were exclusively initiated in the T-cell activation terms like T-cell activation and downstream TCR signaling. So this data suggests the cell type specific regulatory role of the stripes. So in summary, the Stripenn utilized the Canny edge detection to detect the stripes, and it has two scoring systems like p-value and stripiness. And the structural protein like CTCF and cohesins are initiated at the anchor of the stripes, and stripes are involved in transcriptional regulation. And finally, stripes are potentially involved in the cell identity gene regulation. For the future plan, we will discover the formation of a stripe at the single cell level using sequential fish, and also we will explore the difference between loop and stripes. Since we have 4DN Avengers team at Penn, I expect that we will make a great advances in this field. Finally, I'd like to introduce how Stripenn is installed and run. Stripenn can be run using this simple command line, and you can run Stripenn using this simple command line. And this is an output. Output is a simple text file that includes the coordinate of the stripes and the score of the stripes. And it can be directly uploaded to Juicebox to see the stripe codes. And finally, I'd like to thank all of my lab members and collaborators. Thank you for listening to my presentation.